The overall goal of this method is to effectively direct the differentiation of pluripotent stem cells into retinal pigment epithelial cells in a homogeneous layer.
This method helps answer questions in the field of retinal and eye research, such as how the retina develops and how to establish a stem cell disease model. The main advantage of this technique is that it's faster and more efficient than spontaneous methods.
This method uses a combination of growth factors and small molecules to direct the differentiation of stem cells to become immature RPE in 14 days and mature functional RPE after 3 months. Demonstrating portions of the procedure will be Cassidy Arnold, the Associate Director of the Laboratory for Stem Cell Biology and Engineering at UC Santa Barbara.
To begin this procedure, grow stem cell colonies in feeder-free, serum-free conditions to approximately 80% confluence before passaging. Then, coat a 12-well plate with extracellular matrix-based hydrogel as per manufacturer recommendations, and allow it to set for 1 hour at room temperature or overnight at 4 degrees Celsius. After that, aliquot the volume of retinal differentiation media and PBS without calcium or magnesium needed for day 0, and warm them in a water bath to 37 degrees Celsius. Next, add the growth factors to the warmed RDM and bring EDTA to room temperature.